Welcome to The Explainer. Today we're diving into something that sounds like it's straight out of science fiction, but it's happening right now. We're talking about the incredible hunt for the earliest signs of cancer, not with a scan or a surgery, but with just a single drop of blood and the power of artificial intelligence. I mean, just think about that for a second. What if your next annual checkup included a simple blood test that could spot cancer when it's just getting started at its most treatable stage? This isn't some far-off dream. It's the very real goal of this revolutionary field known as liquid biopsy. So, where's the magic? Well, it's all happening right inside our own bloodstream. You see, our blood is filled with these tiny floating fragments of DNA called cell-free DNA, or cfDNA for short. They're shed from cells all over our body. Now, if someone has a tumor, that tumor also sheds its own DNA into the bloodstream. And that's the jackpot. That's the circulating tumor DNA, or ctDNA. Think of it like a biological crime scene, and the ctDNA are the fingerprints the tumor leaves behind. But here's the catch, and it's a big one. Those cancer's DNA fragments are incredibly rare. Finding them is like trying to hear a single whisper in the middle of a roaring stadium. So, to pick out that whisper, scientists are teaching AI to become a master linguist, fluent in 3 secret languages hidden within our DNA. Okay, so the first language is the most obvious one, right? It's the genetic code itself, the actual sequence of our DNA letters, the A's, C's, G's, and T's. AI models are trained to pore over this code, looking for those telltale mutations, those little genetic spelling errors that are the classic hallmarks of cancer. What's so cool about this is how they're doing it. You've probably heard of AI models that can understand human language. Well, they've adapted that same idea for genetics. A model like DNA BERT doesn't read sentences. It reads what are called k-mers. Think of them as short, overlapping words made of DNA. By analyzing millions of these words in context, the AI learns the grammar and syntax of our genome, which makes it amazingly good at spotting when a sentence just doesn't look right. And this approach is a total game changer. By just having the AI read the entire human genome, all 3 billion letters, it starts to recognize important biological patterns all on its own. It learns to spot things like gene promoters or binding sites without being explicitly told what they are. It's like teaching an AI to read so much Shakespeare that it just intuitively understands what makes a sonnet. All right, clue number 2. This one's a little more subtle. It's not just about the words in the DNA, but about how the book itself is packaged and even torn apart. You see, cancer cells package their DNA differently from healthy cells. So when that DNA breaks up and floats into the blood, it creates a very unique signature, a distinct pattern of fragment lengths and locations. And just look at the incredible scale we're talking about here. To really understand those patterns, you need a wide-angle view. Models like DeepMind's AlphaGenome are absolute powerhouses. They can analyze 1 million DNA letters all at once. This lets the AI see the entire genetic landscape, not just a tiny piece, giving it the full context it needs to spot those cancer-related fragmentation patterns. And then there's the third language, epigenetics. The easiest way to think about this is to imagine little chemical post-it notes stuck all over the DNA. These notes are called methylation tags, and they basically act like on-off switches for our genes. In cancer, these switches get all scrambled up, creating a very clear, measurable pattern that the AI can be trained to recognize. Okay, so we have these 3 different kinds of clues: the DNA sequence, the fragment patterns, and those little epigenetic sticky notes. By themselves, they're pretty powerful. But the real magic, the real breakthrough, happens when an AI can act like a master detective, combining clues from all 3 languages to build a completely airtight case. So let's meet one of these master detectives. It's a model called ELSM, and it's one of the most advanced out there. What makes it so special is that it was designed from the ground up to be a multi-talented expert, specifically at fusing together all those different subtle clues hidden in the DNA fragmentation patterns. So how does ELSM do it? Well, it's got this really clever 2-stage process. First, it pulls in more than a dozen different types of fragmentation clues from a blood sample. Then, it uses something called an attention mechanism, which is a fancy way of saying it acts like a seasoned detective. It intelligently weighs all the evidence, figuring out which clues are the most important for that specific patient's case, and then it delivers its final combined prediction. And that's the absolute key takeaway here. This isn't some generic one-size-fits-all test. By By personalizing the analysis for every single blood sample, the AI can zero in on the most relevant clues for that individual. And as you can imagine, that makes it dramatically more accurate. Okay, that all sounds amazing on paper, right? But the real question is, does it actually work? How well do these AI detectives really perform in the lab? Well, the results are, frankly, incredibly promising. So, in rigorous testing, the ELSM model scored a 0.972 in something called area under the curve, or AUC. Now, to put that in perspective, AUC is just a measure of a test's accuracy. A perfect score is 1.0. A score of 0.972 is, well, it's almost perfect. It means the model is exceptionally good at its one job, telling the difference between a blood sample from a person with cancer and a person without. And this isn't just a one-hit wonder. Another massive study that combined fragmentation patterns with genetic features got it right over 87% of the time. But just as importantly, it correctly identified healthy people almost 98% of the time. And that's absolutely vital, right? You need that accuracy to avoid causing unnecessary stress and follow-up procedures for people who are perfectly healthy. And this table really lays it all out. It shows you there isn't just one way to solve this mystery. You've got models like ELSM diving deep into fragmentation, others combining that with genetics for high sensitivity. Then you have commercial tests that focus on those methylation post-it notes to get extremely high specificity. Each strategy has its own strengths, but they're all pointing to the exact same conclusion. AI is an unbelievably powerful and versatile tool for finding these hidden cancer signals. So where do we go from here? What does the road ahead look like? And what's standing between this incredible technology and your local doctor's office? Well, before this becomes routine, there are definitely some big hurdles to clear. The number one challenge is still sensitivity, getting even better at detecting those microscopic signals from a stage 1 cancer. Then there's the black box problem. We need to make these complex AI models transparent enough for doctors to really understand and trust their decisions. And of course, we have to make sure these tests work equally well for everyone by training them on data from all kinds of different populations. But even with these challenges, the fusion of AI and genomics is pushing us closer and closer to a world where finding cancer early is truly as simple as a routine blood test. It really feels like it's no longer a question of if, but when. And that leaves us with one last question to think about. How soon until your next physical includes an AI-powered cancer screen?